Going on the assumption that people can hear me. Welcome in this room at the Institut Pasteur in Paris. Welcome to you, everybody out there, for joining us today. I'm introducing briefly this event, which is a scientific conference in the context of the TranSYS Data Analysis Winter School. This winter school takes place from the 24th today to Friday here in Paris locally and internationally online. And it's part of the TranSYS school. TranSYS is a Marie Skłodowska project of the European Union that's funded from 2019 to 2023, and its main objective is to train a group of 15 competitively selected PhD students in 24 interdisciplinary teams in Europe in the area of personalized medicine. And the idea here is really to get more than the sum of the parts, namely to have students experience 3 different elements of personalized medicine from some very basic questions in personalized medicine to which are about understanding and disease at the molecular level, etc., and doing some preclinical science over clinical trials that happen to test potentially new treatments, and then do the translational science that comes with that. TranSYS includes 12 partners, academic, institutional, and corporate, and 6 additional corporate organizations and 6 degree granting institutes. Today, we greet you from here at Institut Pasteur, which is an international scientific organization, is in the heart of Paris. It's got a bit of history. It's been founded in the 19th century. Today, we are here on a campus of 55,000 square meters with 12 scientific departments, 134 fairly independent research units, 20 technological platforms, which are of course key for personalized medicine and understanding disease at the molecular level, 2,000 odd employees, 1,200 PhD students overall from 68 different nations. Some of what we take for granted today and that we use for trying to develop personalized medicine has been developed here. I don't know whether it's this building. In the building in which we find ourselves is named after Elie Metchnikoff, for instance, who figured out that the immune system works by and kills pathogens by engulfing them, the process of phagocytosis. Another tool that we use in personalized medicine is to try to understand and separate molecular processes by their transcription. And it was on this campus that people figured out that what the process could be for gene regulation, or transcriptional regulation, and that is actually encoded in the genome. So around here, Jacques Monod and François Jacob figured out that there's actually a protein that whose function it is to regulate the production of other transcripts. So without much further ado, I would like to then open the meeting for the first session. Just before that, I want to actually show you this photo of the 15 students that we took yesterday, who are all here in the room. You can see them online right now, but they're all here. And I'd like to pass it on to open our wonderful program to Damiana Rosman, who joined. He's another partner in this program, who joins us from Ljubljana, but in person. And she's going to be the chair of the first session.
So thank you, Benno, for introducing the session. Dear students, dear colleagues through the web, it's my pleasure to introduce the first speaker today. This is Professor Alexander Skupin from University of Luxembourg, and his research expertise is in spatial systems biology / systems medicine. So he will be talking about single cell analysis, and his focus of research is more in the neurobiochemistry or neurobiology. Alexander, the screen is yours. Would be nice if we can see you in vivo, but we're looking forward to your lecture, please.
Yes, hello everybody. Yeah, first, I actually also would love to be there in person and to have more fun with you. Unfortunately, I am still too involved in the local COVID scenarios here, so that's why I could not join as much as I want. So anyway, hello everybody, thanks for joining, and my warmest regards to Paris, and I hope, Benno, that I will also come and visit soon, latest after the current wave. Okay, I will talk to you now about single cell modeling, so why it's useful, what can be done, what cannot be done so far, and how we have to think about this new challenge, and also to include a little bit of modeling to get a better understanding about the regulations that Benno just mentioned, which is visualized here in this epigenetic landscape gimmick from Vatican that we will see again during the talk. So the outline is a little bit that I will give first the motivation, a little bit of brainwashing for the PhD students. Then I will talk about single cell analysis. And I think it's really important that you know what kind of data you're dealing with. So that's why I will give a little bit more details about the different experimental approaches that are out there with their pros and cons. And then we'll exemplify the steps of data analysis and single cell analysis with one concrete example, and then add there a little bit of the questions that are open. And subsequently, I will then show why modeling, so mechanistic modeling, can be actually an important step to generate mechanistic insights. And then I will try to conclude with some takeaway messages. Okay, so let's start with the brainwashing. So as Benno introduced P4 medicine, so the vision of P4 medicine is to predictive, preventive, personalized, and participatory. And that is the vision of Lee Hood, that we are all brainwashed with, and so we try now to pass the brainwashing on to you. And the idea there is really to make medicine or bring medicine to the next level. So in the past, medicine has been rather intuition driven. So it was a doctor who, based on his experience, then found the right treatment. Currently, medicine is also evidence driven. So meaning, so you know, from clinical trials and drug screens, so what is actually could be actually a mechanism, but very often this is also pure statistics. So where the vision for P4 medicine and precision medicine is actually that we have such a good understanding what's happening in the body, so that we can actually target the issues of health in a very concrete and specific manner. So as you know from your everyday experience, or hopefully not everyday experience, but if you go to the doctor, we are not there at precision medicine, and there are only a very few showcases where actually precision medicine can be done. And the reason for that is that we are still not so good in our understanding between about the relationship between the genotype and the phenotype, meaning that we need actually a better understanding about the phenotype, and that requires, of course, more data. So then, if we would have such more comprehensive understandings, the mission and perspectives of P4 medicine would be, for instance, so to have a digital twin. So meaning you have an in silico representation of you that can be then used to test, for instance, different drugs. So given your genome, your genomic information, plus your phenotyping, one could then make better targets for drug treatment. To some extent, this is, for instance, already done in cancer therapy. So where if you have a specific mutation, so then based on statistics, so there is a recommendation which chemotherapy, for instance, is more applicable for you. But the idea of this digital twin is also then to have this preventive aspect, so not only to optimize the urgent treatment strategy, but also to give a recommendation what, given your genome, would be a good lifestyle for you. So for the deep phenotyping, so we of course need much more insights into the regulatory mechanisms. So, and one way to do that is now this organoid technology, so where you can now take patient cells and then generate iPS cells from which you then can reproduce with the right protocol different organs, for instance. And this is what I gave in the kickoff meeting, so an overview about iPS cells. But in general, so this is a still coming up technology, maturating technology that will hopefully enable us to actually generate in vitro models of patients and subjects. Oops, this was one too fast. For this precision intervention, what we then actually need, and that's why this personalized model systems can be rather useful, is that we need a better understanding about the specifics of a specific patient. So, and that is, for instance, important to bring regenerative medicine on the next level, so that we understand how to regrow tissue or, well, suppress tissue in cancer, for instance, and also to identify actually the cells of concerns. So I mean, like, again, the example of cancer. So there's this this concept of cancer stem cells, and these start with a very, very small amount, right? So meaning that actually it's very often that we have to understand and identify rare cell populations, and you see already there. So this is actually a major driver for the need for single cell analysis. So then how to deal with this challenge, and there's systems biomedicine is then in a similar manner as systems biomedicine has these two complementary approaches, where you have on the right the top-down approaches, which is based on high throughput data, so where you can apply omics or high content, high throughput imaging to really have big data of your system. So like your organoid at the moment, later on, hopefully in a standard way, also from your body, so directly subject based, and then. And that's the main topic here of this workshop. So the challenge is then to go from this big data and integrate this towards a more mechanistic understanding, so that you really can identify potential issues, and then to go into the other direction. So when we have identified these key players, then the bottom-up approach can actually, on the one hand, give us a validation, but on the other hand is then also the tool to actually push the system into the right direction. So meaning that here it is the synergy between top-down and bottom-up approaches that we can use from systems biomedicine. So in terms of the multicellularity that we are living in, so meaning the question, how does our body generate this more than 200 different cell types from the same DNA? So we have to understand the cellular diversity to then actually understand what's going wrong if cells take the wrong route and then, for instance, becoming a cancer stem cell, or in the case of neurodegeneration, start to dying, so that neurons dying off. So, and for that we need a better understanding actually about the cell fate. So how does cells decide on their fate? And that again is very often happening or starting in the rare population of cells like cancer stem cells, but also in neurodegeneration, for instance. So in the start, it's only a small amount of cells that getting the problem. And to identify these and then identify the underlying mechanisms there is actually essential to get a targeted and precision tool for better treatment. So, and of course, so that is then directly linked to the need for high throughput single cell analysis and then an integration about the data. So, speaking about integration, here you see again, so that is epigenetic landscape scheme. which is then linked to this differentiation protocol. So then that was his main motivation for his scheme. So to understand how a stem cell or a potent cell is then deciding into or differentiating into a specific cell type. So where the idea is that you have your cell as a mammal there in a valley, and then during differentiation, the mammal is rolling down the valleys and ending up into such an attractor, which then correspond to the cell state or the cell type. So so that's what is going, or this is what's happening during development, and then also later on for regeneration of cell types. What we used or what we are using in iPS cell technology is that we can, or have this magic chemistry, we can now also de-differentiate cells, so and bring them back up into a multipotent attractor, from which we then can start the differentiation into another valley, so meaning into another cell type that we can then use to investigate a specific subcell type, for instance, to understand what might be causing a disease. Another direction in this epigenetic landscape is also then transdifferentiation, so which has again two sides of the medal. So where in disease conditions, so this transdifferentiation can be, for instance, so the development of cancer stem cells. Sorry, cancer stem cells. But it is also a holy grail in regenerative medicine, where the vision would be so that, for instance, in neurodegeneration, so sorry that I'm primed here, in neurodegeneration, so that you actually can replace degenerated neurons by transdifferentiate other cell types like astrocytes that are already there into then the population of neurons that might die off. So so meaning on the schematic level, everything is solved, but actually in terms of bridging there to reality is still not as easy as we would like. So meaning that this is strongly linked then with the question, so how can we understand these mechanisms on the level of gene regulatory networks, which would give us then actually the tool to also start to make precise modifications in this transition, and then with that potentially can get rid of cancer stem cells or regenerate died neuronal populations. So and to do that, to understand this, and then actually to get there, also good understanding about these rare populations. So there is since now 10 years intensively, there is this field of single cell sequencing and single cell analysis, where you can see here the main motivation is that if you have your tissue and you go with a classical analysis, so the bulk analysis, so you then get kind of an overall picture of your tissue, but very often, so as that's well, issues with your health start with rare populations, so meaning that then the single cell analysis, you are able actually to identify the different populations, and then also look into subcellular subcell type specific problems that you can then address. So the advantage there is also that each cell can be then considered as a replicate, so meaning that you then can actually reach a high statistical power of your studies. But, and this is what we will see later, so single cell data comes also with an intrinsic amount of noise, so which is then kind of reducing the statistical power, so meaning that if you have an experiment with 1,000 cells, it's not the same quality, data quality, than if you would have 1,000 bulk analysis, because there you will have a higher coverage, but I will come to that. Okay, so as said, so for the last 10 years, there is this gold rush of single cell methods, so which is here summarized in this review by Svensson and Teichmann from 2008, where you see here the timeline, and here you see the number of cells that were considered in the study, given the different approaches. So, and again, so we will go there a little bit more into detail into the main different approaches that are there, but you can see that so since 10 years, so we have a more exponential growth in the number of cells, where we now maybe start to saturate, but it's more or less a Moore's law of cell numbers, where SmartSex was one of the first methods that was actually then also copied or adapted by others. And so you can see already here, so there is another version of SmartSex and came out two years later, and actually this year there's SmartSex 3 out that is then probably further developing. Another game changer has been Dropseek, so as I will explain later on, and then probably for popularity and yeah, so presence of the single cell stuff, so the 10x genomics approach, which is now the commercial version of droplet-based single cell analysis, is pushing the field even further ahead. And what is not covered here in this review article are the newer developments, like for instance there is the SC-ColorSec publication from this year that is actually then looking into long reads of RNA sequencing, and spatial transcriptomics is the method of the year according to Nature. So here is another overview to show a little bit what are the different approaches based on. And first there comes the art of cell isolation. So somehow to have your analysis at single cell resolution, so you obviously need to isolate the cells, and then there are the classical ones like for instance FACS. So everybody knows how a FACS works. So I mean, in a FACS machine, so first in cell sorting, so you isolate cells into a droplet, and then if you have a marker in this in your cells, the fluorescent marker, so you can then sort the cells into different tubes or wells by adding a charge here. So meaning the droplet in which the cell is isolated is charged, and then you have here a capacity with which you can then push the droplet in one or the other direction. And the other, another approach, and I already mentioned that, so is these machines that are actually generating these droplets in a higher throughput manner than FACS can do. And well, this is a topic of the Dropseek and Chromium that I will speak a bit, and then there are also complementary approaches that are using cell labeling in sometimes even in the tissue. So like in Splitseek, that I will also cover a little bit later. So then you can see here there are then further developments like the droplet generation, and then high density plates for higher throughput. Here you can actually then see a summary what might be here interesting, and so the circle stuff are the things that we will have a closer look to. And what you can see here is also the price column in this thing here, where you see that SmartSeek is extremely expensive, because what you do is more or less like you do an RNA-seq as you would do for your sample. So that's making this so expensive. But on the other hand, you get a high coverage. And so the Dropseek approach is a cheap one, and Chromium is unfortunately still rather expensive. Also, well, maybe there will be also a change with that one. And I will briefly mention then just further derivatives of these technologies. So one is Sightseek, and the other one is to give you a very quick insight into spatial transcriptomics, which is now a methodology that will come more and more. So, and as I was introduced to be interested in spatial systems biology and biomedicine, so that's obviously something that I'm looking forward to. Okay, a quick run through the different single cell technologies. So the SmartSex, as said, was the first approach that actually was able to do not super high throughput, but high numbers of cells with a very high coverage. So what you do in SmartSex is that you use this fax machine to isolate individual cells, and then you lysate these individual cells in the wells to get the RNA, and then you get an oligo-DT into each well, so that is then generating your the template, and by adding here a non-template, say C basis, so you actually then get a functional or PCR ready piece from which you then can generate cDNA. So then the cDNA can be thrown into, or can be is amplified and then is thrown into a sequencer. But as said, so here you really have to run individual cells, or you have to generate a library for each individual cells. And so that is making, on the one hand, gives you full-length RNA sequences, so which is great, where you can also then look into splicing variants that can be used for estimate for maturation and so on. But it comes also with a very high price. So, and actually, when I only did this SmartSex protocol once, because of the price, and you can see here, so those days it was actually even more, it was 15 euro per cell, which then ended up with, where I then ended up based on the budget with less than 100 cells, which was not really like a major breakthrough, where you then do not have the power of the replicator of cells. A game changer in this whole field of single cell analysis was then the Dropseek approach. So in 2015, Benno and co-workers. And the trick here is that you actually have a smarter way to isolate and to identify your cells. And that's based on this microfluidics device you can see here that we're also generating in the lab. And what you have on this microfluidic chip, so, and actually here you see we have 6 Dropseeks on this microfluidic chips, and each has 4 outlets. So, and in one you then get the beads that I will explain in a sec, and then you have oil coming from the other channel, and then the suspension with your cells in another one. So, and then you can see here a microscopy picture about the droplet generation, and this is then generating more than 10,000 droplets in a minute. And in these droplets, we then isolate individual cells and this microparticle, which is actually a bead with a unique barcode. So, and in the early days, one had to generate these beads still by yourself, but so you can now you can actually buy them commercially, which is a big help, and makes Dropseek still kind of a working horse in the field. So what is happening now in the Dropseek is that you isolate your cells with these beads, and these beads have this poly T tail with which they then capture mRNAs with a poly A tail, right? So this is the way how you then actually capture your mRNAs, and then for each bead you have a unique barcode, meaning that in the end you have for each mRNA that you're capturing, you have the barcode, meaning the identification of the bead. So when you have this suspension of your droplets, so what you then do is that you then do the reverse transcription, so to get the cDNA there leading to these stems, and then you can again use PCR for amplification, where you now have for each transcript, you have on the one hand you have the mRNA sequence, and on the other hand you have this unique barcode. And then with that you can merge everything together, and now by doing 2' sequencing, you get from the one side you get the mRNA sequence, and then from the other end you get the identifier, so this is barcode, and with that you can then actually generate this digital expression matrix where you then have for individual cells the different genes. So, and what you can see already here, but we'll talk about this a little bit later, is that this is also rather noisy, based on, first of all, the general stochasticity of gene transcription, but this is very strongly amplified here by the process, meaning different beads have different efficiencies in terms of capturing mRNA, and then, as you can imagine, during the process you may also have further variability that can amplify your cell or your intrinsic noise in your data. So the limitations of Dropseek is that it's actually a unique kind of a reasonable amount of cells to start with. So where at least 200,000 cells should be considered to get in the end in the amount of thousands cells that you can actually analyze. So meaning that you see already there, so there you have a factor of 100 or 1,000 that you need to put in. The reason for that one is that you want only to have a single cell, an individual cell, and an individual bead here, so in this droplet. And to do that, so you run this whole thing into a low concentration regime, so where you then have actually the Poisson regime, meaning the regime of rare events, where you then actually only get very few droplets where you have a cell and a microparticle. This also means that you will spend some money on just sequencing the beads and not the cells, but still Dropseek is rather cost efficient, as you can see here. So in our lab we can bring this cost down to 30 cents per cell, which is pretty good compared to the more than 10 euros per cell from the SmartSeek approach. So another disadvantage here is that you actually, to get this done, you need more hands-on time. So a classical single cell experiment is actually with Dropseek running for 2 days. So 1 day really for the preparation for the encapsulation, and then bring this on the level of the stems, and then for another day then for library preparation to send off to sequencing. And since there is also this customized dimension and induced variability, so it's sometimes hard to merge data from different Dropseek experiments. So, and this was actually what the famous Chromium 10x approach has focused on. And in the end, it's the very same idea. So you also have a microfluidics device, but this microfluidics device is optimized in a way that you can start with a lower amount of cells, because what they managed to do is to optimize this regime where you get individual cells and individual beads into such droplets. And the other difference to Dropseek of the 10x approach is that you do actually do the chemistry with the PCR amplification already in the droplets, meaning so you do not have only the oil, the cells, and the beads coming in here, but actually you also put in the PCR reagents to actually then run through the PCR amplification. So the good point here is so that they brought down the hands-on time really to 15 minutes to half an hour to actually load your sample. So and then afterwards, so you based on the more standardized chemistry, so you also save some time and some pain to then actually generate your library for sequencing. But this comes then with a cost, with an increase of cost. So here you can see so that at our institutes, in our lab, so currently the typical cost per cell is €1.50. So, I mean, if you have better deals for sequencing, so you can break this down, but still, Illumina, so with the Chromium 10x approach, is making a lot of money. So, and people in the field are also sometimes a little bit frustrated that there is not at the moment not a real commercial alternative to it. A potential alternative to it was suggested by Rosenberg and others from Seattle. So, and this is the Splitseek approach, so which is actually not using any fancy microfluidics device, but is using the cell to generate the barcode. So, meaning what you do here is that you have your cell population, you permeabilize your cell, so and then you split your population into different wells that contains different barcodes. So and then you pool afterwards again, and you make another split, put it into another pool of barcodes, and so on. And by doing so, actually you now use you use a combinatorical explosion by this combination, and meaning independence on the steps, how often you label and pool and label, so you can get or you will generate more, or you can generate more barcodes than cells that you have. And in this way, statistically, you will also then generate unique barcodes in your cells. So meaning that the steps are here. So you add in the first step, you add a first barcode. You then add a handle here, so a connector. Add the second barcode, and you go on until your combinatorics is telling you that you have enough unique barcodes generated to cover your cell population. So so I mean, theoretically, so this actually can bring the cost much further down, so to less than 10 cents. So I mean, they claim even to the level of 1 cent. We tried this method early on, and it was not as straightforward as they claimed. So at one point we then gave up because we had Dropseek running at the moment, so they are in the phase of commercialization. So meaning that maybe with the direct targeted kits that they will provide, this can become a game changer. But so far, I am not super convinced that this is already on the level where it is out of the shelf solution. Okay, then as mentioned in the beginning, so I also wanted to briefly mention the Sightseek approach. And Sightseek is again based on droplets. So where what you do now is that you add an additional dimension to your system by putting a label, an antibody label that has also a Pouillet tail here, so that it can be then captured by your barcoded beads. So meaning you stain your cells with the antibody here, and then again it's the same procedure as before. You isolate cells with a barcoded bead, and then due to the Pouillet tail here, so that this will be also then attached to the bead with a unique barcode. And what you then can get is actually the instantaneous information about your gene transcription, so the number of mRNAs that you have, as well as the counts of your antibody in a cell-specific manner. So in the initial version, so this was limited to cell surface proteins, so which makes it rather interesting for immunology. But in the meantime, 10x has adapted that, and now you can also target cytosolic proteins. And here's just an example of what you can do with it. So this is now an example where you have two different treatments, and you get the mRNA and the protein level, and then what you can do about dimensional reduction, you can cluster the cells and assign cell type to it. So as you can see here, these are all immune cells based on the protein level, and then you can overlay actually the transcripts, so the transcriptional level, and then you see here so that for the CD8 cells, which are localized here in the T cells in the protein level, we also see a corresponding high transcriptional level of CD8A, but also a little bit of subpopulations here that can identify either some activation patterns or some differentiation processes. And as well for the CD4 cells, for instance, so you see this is the same thing here, that you have a good overlay between the protein estimates of your cell type as well as with on the transcriptional level. But we will get to the cell type identification in a bit. So as mentioned before, so Spatial Transatomic was honored to be the method of the year by Nature, and this is, or well, I mean, there are not so many easy approaches to it, and it's actually the only one that is available right now as a out-of-the-shelf solution, where out-of-the-shelf is again a little bit problematic, I would say, is by 10x, so the Vision Spatial Gene Expression. And how this works is that you have a chip or a plate with micro wells here, where you have again primer, unique barcoded primers. You then isolate your cells, or you lysate your sample ahead of this on this plate. Get the barcode labeled here in a spatial specific manner, and then you can use again multiplex sequencing to then get the mRNA count. So as then, and which gives you then the nice clusters. And with this one, you are then able to map the spatial dimension to the mRNA mRNA level, as done here by Faulkner, Carpeaux, and et al. In the intestine development. So here you see the colon development over several weeks. So this is again not as cheap as we would like to have it. So meaning that running such a sample in a spatial resolution, so you are easily above 10,000 K. So meaning it's not there on a high high throughput level, which we would actually need to make really then also time development analysis as well as complementary analysis. So. And one thing that might help with it is then if there are academic approaches to it, and so a promising approach. Here it is. A promising approach is this expansion sequencing exec called. So mainly based on previous work from by George Church. And what's happening here is that you take your sample and you put it into a gel. So here, and then this gel can now be expanded. And with this one, so you expand also your tissue. So. But still, so you have the idea about your geometry, because you do this then by microscopy. Once you have expanded your sample, you now anchor the mRNAs, so by chemistry, so that they do not get flushed away. And then you do the in situ sequencing, meaning that you now add the fluorescent tagged primers here that are binding, oligos, so that are binding to the mRNA, and then you can now, by microscopy, make a sequencing in your sample. So this is visualized here for a mouse sample, how it looks like. So you have here the overall sample, and then you have the zoom into this field of view here with the different markers. So, and then you see here the sequencing round, so meaning that you go now confocally through your sample, and for the 4 different base pairs, bases, so which are then having a color code. And you do that until you think that you have a good coverage, or rather unique coverage for your RNA things that you're interested in. So in the example, they used 17 rounds, so meaning 17 sequencing here. And afterwards, what you do is that you then do an ex-Z2 sequencing, so meaning that you then put all your sample into the sequencer, and eventually you can then map your in situ reads, so your imaging-based reads, then with the long read of your mRNA, and with that one you can then link your spatial location with the transcriptional level. So obviously, so this is not something that you do in an afternoon. Unfortunately, I'm not sure if this will make it to a really high throughput thing, but at least it is an alternative to the commercially available thing, and is also addressing another issue that I haven't mentioned with the 10x Chromium Vision tool, where actually you have your microplates, so with this micro wells, and you lysate your sample above it, but that is, I mean, these wells are really small, and 10x is claiming that this is then single cell resolution, but you will always have like cross contamination, meaning that your single cell resolution is also diluted, is there. So whereas here, this approach might actually be an approach where you can make sure that you actually are only getting the transcription level of an individual cell. There is another protocol out or published like 2 months ago, Dropseek in Nature Biotech by the Louvain-la-Neuve group. So, and this method is not requiring microscopy, so that therefore may be better for high throughput, but I haven't really evaluated that yet. But just to let you know, so there are now there's a zoo out of single cell technologies with different pros and different cons, and it also depends a little bit of the target of the study, which is the appropriate one for you. What all these approaches have in common is so that in the end you get this digital expression matrix that looks rather noisy. So here you see, for instance, now an example for differentiation in the context of Parkinson's disease for genetic controls or a gene-corrected control, and Parkinson mutation. We have here for 4 time points, we got more than 3,000 cells and a coverage of 16,000 genes. But what you also can see is that for each cell, so a cell corresponds here to a column, so you have different coverage. So meaning that for some cells you have a very high coverage, so meaning you get more genes identified than for others. So, and that's actually then afterwards an issue for data analysis and data interpretation. So for those who are working in the fields, the buzzword is then zero inflation, and there are methods out who try to compensate for that. This was rather a hot topic, and so different approaches has been suggested there for the last I don't know, 4 or 5 years. In the end, in practice, actually, so we stopped using that one or mainly we stopped using that one because, so with the increasing coverage, which is linked actually to decreasing costs again, so it's always the money, we can now compensate better for these losses here, and then to be on the safe side to not introduce any artifacts based on your inference of missing values. The general trend in the field is not to do so much inference here. Just as a side note. So still, this is kind of, or was really new data, and we are still not there to make their straightforward one-way solution for the analysis. And one issue there is the dropout that you have. So meaning you, as you have seen, so we have there zeros, this gray values are zeros. I should have mentioned that. Zeros, where the question is, are these now really zeros in terms that the cell hasn't expressed this gene, or is this a technical zero because the mRNA was too low abundant, was lost? Was there a mistake in the alignment procedure, and is there a general, or is there a systematic issue? So I mean, this is also what you sometimes see, is that so for some mRNAs the capture rate is just not good enough. Another issue that is there is cell damage. So specifically, if you run that through Dropseek or also through a fax, so what can happen is that due to the pressure that you put on the cells, so the cell membrane can break, and then you will have leakage of your cell. So meaning that here again the art of isolation is key. And then if you get leakage from your cytosol in the mRNA, you will get a wrong picture. So an evidence that something is going wrong there is if you get an enrichment in mitochondrial mRNAs. So because since mitochondria have their additional membrane, so they are more protected, meaning that mRNAs that are localized in mitochondria are less likely to leak, meaning that cell damage, or a first indication for cell damage, is actually if you look into the expression profile of mitochondrial genes, and if you increase here above 10% for sure, so then you can be pretty sure that this cell has been leaked, and therefore it should be excluded from the analysis. Another approach, or an approach to circumvent that, is then to go not for single cell but single nucleus analysis. And that actually turns out to be, unfortunately, very often the case to do in the brain, because the brain is so dense and compact that it's hard to make there a very clean isolation, specifically for older brains. So you can, or you will induce a high amount of cell damage, specifically to complex morphology cells, leading to leakage and from neurons or also oligodendrocyte, meaning that your whole brain composition analysis is then biased towards more easily isolatable cells. So another issue in the single cell approach is that you have variability, and so there is, on the one hand, there's a variability that is based on biology, and this is the one that we are actually interested in, but then you have the variability that is induced by your technique, by your technical treatment. And then in terms of later on identification of cell types and clustering, so there are also confounding effects like cell cycle. If you're working with non-synchronized cells, cell cycle is a classical co-founding factor that will lead to, or will suggest you, some subpopulations that are actually only different in their cell cycle phase, but not per se in their cell identity. So on the other hand, so I mean, cell cycle is something that can be of high interest in cancer research, right? So meaning that there it's again a question about, so what is the target of your study? And then you have to adapt your analysis accordingly. Okay, so to make this a little bit abstract thing more visible, so I will go with you through mainly the lower part here of the classical gene analysis, where the first part is based then on the classical quality control, so where you look into how many reads do you actually get per cell. So do you have a bias in mitochondrial genes and so on? So that's all coming here after this sequencing quality control. And then you do the normalization and look into co-founding factors like batch effects, and then you can start with the meta-analysis looking into clusters of cell types, differential gene expression, and so on. So as you can see here, so the number of approaches have also increased, similar to the number of experimental protocols, or the number of cells for the different protocols. And there are mainly 3 tools or 3 platforms that are efficiently used. So mainly it's R tools, then there is Python, a collection of Python tools, and also some approaches in MATLAB, but I think the field is really focusing on R and Python. So meaning if you start to, or if you want to use some predefined packages, I would recommend you to go with R or Python. And in terms of getting a little bit of a standard in the field, so there's the Syrup package and the Monocle package in R and ScanPy, which are so now the working horse in the fields. But as said, so there are additional tools for specific analysis that might be rather useful. And then what we are also always doing is that we do actually a combination. So very often in the R environment, sometimes we use ScanPy, which is written in Python if it's really large data sets in terms of computational efficiency. But as a good starting point, so the Syrup and the Monocle packages are good tools. Okay, so now I want to run you briefly into through an application that we're doing in our lab to give you a flavor about the issues and the potential that single cell analysis has, and what the data that we have is actually based on brain maturation. So it's specifically focusing on this gestation of the brain here, which happens between week 12 and 24. And what we have there is, again, because it's brain, so we will have here not single cell but single nuclei data, and then to have good coverage. So you then cannot use actually Dropseek, because the coverage of Dropseek is not, or you can use Dropseek, but it's better to go then for 10x data, because with 10x you have a better coverage, a higher resolution, which is actually needed to then get a better picture of the small amount of mRNAs that are localized in the nucleus. We have then 4 time points. So it's for the later stage of gestation here. And we were looking into 4 different brain regions, the cortical plate, striatum, thalamus, and hippocampus, where a motivation for us was to look into the striatum and the hippocampus, which are linked to neurodegeneration. Hippocampus for Alzheimer's, the striatum is involved in Parkinson's disease, and we know that this early phase here of brain development is also a vulnerable phase for schizophrenia. So, and therefore, specifically striatum as well. So that's why we focused on these things. And here you see again the workflow, or a workflow where we have here the different samples that has to be merged. Then we address the potential batch effects by Liger, so which is a good tool if you're working with this kind of data. So my recommendation is to look into Liger, and then we apply pseudo-time projection, which I will explain in a bit, and looking into enrichment of cell types and gene expression to then get a better perspective of this maturation behavior. So, and another thing that turned out to be rather useful on our side is to really organize these workflows by SnakeMake, so which allows you to actually take track of your pipeline. And based on painful experience, we also now actually do Dockers, so that we for a paper that we just submitted, we really make a full package of all applied tools, because if you get then a small update of one of these packages, so your clustering and so on can change slightly, but in terms of reproducibility, so it is still highly recommended that you actually get as much as a frozen perspective as possible. Okay, so as said, so we have 4 time points and 4 brain regions, and what we have used here is a hashing approach. Where similar to the Sightseek, where you add this antibody to your cells. So we actually used here also an antibody, so a primer to barcode your sample, right? So, and then an advantage of this barcode is that we now each sample has a specific barcode. And now when we merge these samples and then doing the 10x analysis, we also get a hand on the number of doublets. So what is a doublet? A doublet is now a droplet in which you have not an individual cell, but where you have 2 cells or even worse, 3 cells, because then these cells will have or will get the same barcode from the bead, right? So, and then this is called doublets. And we don't want to have doublets because in the end you get then neuroastrocytes or an epithelial cell with a neuronal marker, which is not something that you can actually claim as a new cell type. So now having this sample unique barcodes here, you will then see that after your sequencing, meaning that what you would expect is for the samples, for clear separation in the barcode of these barcodes, right? And all this mixed population is then based on doublets. So, and obviously, so we can resolve the doublets between the different labels, but it also gives you then an idea about the percentage of doublets that you would actually expect also for the same barcode. Okay, so what you see then here is the identification procedure of this hashing or of this demultiplexing. And again, so if you look here now into the dimensionally reduced space, you can see here the 4 sample plots where you then get this doublets that correspond to mixed barcodes. So by doing then this demultiplexing, so what you can see here for the different samples, so that we still that we get only a rather low number of doublets. That is good. So for the hippocampus a little bit more, but still we are there in the range of below or around 10%, which is something that is kind of expected. So for the 10x approach, you can reduce, so as a side comment, you can reduce the doublet with the Dropseek approach, but then it comes with the need of higher cell numbers, so that you need. So anyway, so for the sample, we have now more than 50,000 cells, which are actually nuclei, with a rather good coverage. And then the first exercise that you have to do is then to look into the batch effect. So, and to do that, so as said, we apply Liger, the Liger tool that is using regression models to understand if there is a generic bias between these samples. And in this study here, since we had our hashtags, so our labeling, our barcode labeling, so we have also for each sequencing sample, we also have then the mixture of the population in there. And with that one, you can see that we do not have a strong enrichment after batch correction for any of the samples. So it's a rather unique coverage. And also, if we now break this down into brain regions, so we do not see anything outstanding. So, I mean, here again, so this is where you really have to have still a close look on your data. So there are, as I said, Liger is doing a good job and is also providing some quality controls tools for that. But it's still kind of, you have to understand the data and to make sure that you do not end up with the co-founding factor. Like, for instance, here in this case, if you look into the cell phase and the cell cycle phase, you see that there is actually different, so that some population correspond to driven by the cell phase, that you can then afterwards also reduce this Liger. So I mean, there are now available tools in a plug and play mode, more or less, that can reduce the co-founding factor of cell phase, but as said before, depends on the question that you're asking. And as another investigation that you want to see is that you do also get not a very weird picture in terms of your duration, the age of your sample. So we're obviously, again, so it's a question about the question. So obviously, so for maturations, for instance, so you would actually expect that some subcell types at least are more pronounced in a later stage than in an early stage, and vice versa. So, but again, so if you would only observe for a specific time point one strong cluster, you at least should check if this is not an artifact. So, meaning that's the first step to make sure that your data is kind of consistent and as unbiased as possible. So now with reducing these batch effects and also here the cell cycle, so we can now map the data onto a UMAP space. So, and I mean, again, so historically, if you can use this word in this context, so in the beginning, we were all using PCA or ICA for dimensional reduction. Which has the advantage that you can still have a clear understanding or an interpretation of your dimensional reduction. But with the increasing complexity of the samples and the higher coverage, so it's getting then really hard to get a good separation, a visual separation that you then can show, which is then kind of still the aim to have clear separation that you can then assign to cell types, right? So that's why after PCA, ICA, there has been then the TISNI mapping, and the current tool used in the field is mainly the UMAP mapping. So just to let you know, if you see that different axis, so it's also probably you can make a guess about the publication here when you look into the embedding space, the dimension of the embedding space. Anyhow, so this is now the finally quality controlled, unbiased data set and the mapping in the UMAP space. And then what you then use in the Surat packages, they're already rather good to give you cell type markers. So you can now assign a cell type to these different populations. Where we have here mainly the excitable neurons, and here you see interneurons, so the inhibitory neurons. Then you have here some, again, GABA is obviously also an inhibitory neuron population connected to reactive glia cells. You see OPC, so oligodendrocytes, the microglia, and so on, which is all then based on some cell markers that you can either use from Surat package, or there are then other databases out in the field. So having this data, we can now make a first look into if there's any brain region specificity. We have a stronger or a higher presence of some populations. For the time points, we also see a trend towards the borders for the week 24. One thing that I should have started with, I just realized now, is that another quality control thing is actually that you look into the coverage of your cells. So meaning what you see here is now the number of features that can be extracted for each cell based on the sequencing, which is to some extent the number of counts, so the number of mRNA reads that you get per cell, and the number of genes that you identify. And then there are different ways how to combine these things to get feature score for it. And again, what you want to have here is that you want to have not a very, well, you want to have a rather even distribution, so which is then also a quality measure. Okay, now I have to go be a bit faster to finish in time. What you then can do, so we have, you see here now the different cell types that could be identified in our samples, where we have here the excitatory neurons, and then here the inhibitory neurons. And from here you can already see, so that you have in the cortical plate you have a higher contribution to excitatory neurons, whereas in the thalamus and also to some extent in the striatum, the interneurons are more pronounced compared to the excitable neurons. Yeah. So then, as next step, what we are typically interested in is, so what is the difference between the different cell clusters that we have, that can be then also linked to either to cell types. So what is again here the method? So you can see here for each identified cell type cluster, you see now here is the most differentially expressed genes named here, and then if you map that onto your UMAP projection, you can then get another identification of your cell type. So SOX9 here for inhibitory neurons, so here a marker for excitatory neurons, and then depending on the specific questions that you have, so you can then zoom in and get some also kind of into the subclustering. So, and if you have some specific target genes that you're interested in, maybe potentially you stumbled on it by another approach. So then you can also look how does these genes are expressed in dependence on the cell type. And here you see, for instance, something expected that GAD1 is as a GABAergic marker highly expressed in the interneurons. So as you would expect, where these genes here correspond to the excitatory neurons. So something that is now becoming more and more popular is that you try to combine also different data sets. So in our case, so there is one data set out that is also looking into the brain development at a similar time point. And what you then can do is that you integrate these data sets on ours. So this is now in color. You see the data of Pouliadakis from 2019, only at a snapshot. And in gray you see our data, and then you see that actually there's a rather well overlay where we actually identified some more cell clusters. But in any case, if we then look into the cell identification of our cells compared to the previous cells, then you see based on the diagonal, so that the Jacquard index as an indication for similarity is rather high and rather well, so meaning that our cell identification, cell type identification maps with independent data as well. Something that is then interesting in terms of also going into medical direction is that you can actually make use of GWAS study by Magma. So it's a tool by Watanabe et al. From 2019. And the idea of this tool is that you have a GWAS, so a genome-wide association catalog, so where you have your risk lookie identified, so given a specific disease, and then you can now look into your data, so which of these risk scores are associated to which genes, and are these expressed in a cell type specific manner. So and then Magma is using a regression model to then generate a cell type specific risk score. And just as a quick example, so this is now based on our data here. given the different cell types that we have identified, and what you then can see, for instance, that for schizophrenia, so which is these GWAS studies here, so we see actually that we have some rather strong impact for more or less all cell types, but also for the OPC, so for the androcyte precursors in the striatum and the hippocampus, what is would actually be expected. So the color scheme is not so well actually. So it's the highest risk score is actually here, but still, so the GWAS catalog is not as sensitive, I would say, so to really break this further down, meaning that here we would say that it's a rather cell type independent risk factor where only endothelial cells are not having any of these neurological disorders. Something else that was interesting, and what you can then deduce from this one, is that if you look into bipolar GWAS studies, or if you link the bipolar studies, so that it's mainly the hippocampus that is actually taking their risk factor. So, I mean, again, so this has to be always taken with a grain of salt, because this method is only as good as the input data. So, which is then in this case here, on the one hand, the GWAS resolution, and on the other hand, the single cell data. But anyhow, so just to let you know, there is data, or there are tools out that also support this transition from your data into cell type specific risk score identification. Something that we were then interested is, and now I come to the idea about the subclusterings, that we were wondering about the neuronal maturation. And for that one, you can see here again only now the neuronal subpopulation with the excitatory branch here and the inhibitory branch there. You can then again run differential gene expression and actually see that there are blocks that are shared by the interneurons and blocks that are shared by excitatory neurons, and that there are then the cell types specific differentially expressed genes. So in terms of the maturation, so then we are coming to entering the field of pseudotime, where you now try to estimate again by regression models. So what is the directionality of this maturation, and what are branching points, right? So this branching points in differentiation. So here you see now the lineage estimates generated by Monocle. So I mean, there are also other tools out there. Again, from our experience, Monocle, specifically with Monocle 3, it's a rather robust and adaptable tool. So meaning that if you want to go in this direction, I would recommend to use Monocle. What we were able to do with Monocle is to see here two lineages associated to the excitatory neurons. So starting here from the less mature to the more mature maturing neurons. So, and then the other direction of the inhibitory neurons, the lineages 3, 4, and 5, that you then also can link to by GO-term analysis, where you see here classical classical things or classical behavior pathways for excitatory neuron, including axon guidance, cholinergic synapses that are an issue in Alzheimer's. And for inhibitory neurons, you see then also corresponding pathways that you would expect in this lineages parts. So meaning that's nice. We get an idea about the maturation, but in terms of transition points, all tools out there are not very specific. So therefore, I personally believe still in the analysis that we have established, which is based on the attractor picture. So meaning the idea of this transition index here, which is relating your gene-gene correlation to the cell-cell correlation, is the following. So that if you're in an attractor state, so meaning your cells sitting all in a valley, so your cells are rather similar. So therefore you have a high cell-cell correlation. And on the other hand, you do not have a large spread of your genes, because so this, the system is in equilibrium, right? So you have only major movement. Whereas if you go into the transition, so if you pass a branching point, right? So there you have an induction of the change in the gene regulatory network, and then also a larger cellular diversity. So, and if you apply this idea to now the different neuronal lineages, so by calculating the cell correlation, the average gene-gene correlation, we then actually see these transition points in the maturation for all these lineages. So meaning here really, so the potentially a two-step things for the excitatory neurons, and then this transition towards the more mature and stable inhibitory neuron state. Okay, but what does it tell us about cell fate? And so the underlying mechanisms there, and since this neuronal differentiation is rather complex, I just want to briefly mention an experiment that we actually used to develop this transition index strategy, which is blood cell differentiation. So we started here with hematopoietic stem cells, used an EPO or immune cocktail to either induce the erythrocyte or the myeloid lineage. And then from the single cell data, we could then see how actually this differentiation procedure is happening, first by destabilization of this stem cell attractor, and afterwards a sorting out of such a maturation into the corresponding cell types. And I think this I've shown already in the kickoff meeting, so there that based on this data we can actually now quantify this epigenetic landscape, so making this abstract gimmick from Wellington into animated dynamical behavior. So then again, similar idea as before, we calculate cell-cell and gene-gene correlation, and then we could look, or we can quantify this transition of the differentiation by this transition index. Here, and we see that at day 3, actually, we have this strong signal in this transition index, meaning so that it's becoming clear that there is something happening. And if you look into the PCA space in this potential for the different treatments, you see particular for the EPO treatment for inducing the erythrocyte lineage, you see that at day 3 we have cells that are actually going into the wrong direction. So we called them rebellious cells and gave the hand-waving explanation in the paper, so that this is based on this bifurcation, so on this transition characteristics where in the attractor state of the stem cell, so cells are close by, so also having small critical index, and now by inducing the transition towards a lineage, so you destabilize this attractor, and then by the heterogeneity of your stem cells, one of these cells end up into the wrong potential. So meaning that you end up then with these, as we call them, rebellious cells. So this is a nice, was a nice hand-waving explanation. But what was bothering me is so that we never explain why these cells disappear later on. So, and my co-workers then always claim, yeah, well, they're just dying off, blah, 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 blah. But I wanted to get a little bit more of a mechanistic understanding of that one, and that is then briefly linked for the last four minutes. How we can use actually the other ways of the bottom-up approach for a mechanistic insight, where modeling cell fate has to consider the intrinsic noise, the multi-stability, so it means the different cell states, and the induction of cell fate, plus the multi-scale organization. Meaning that this description needs a probabilistic ensemble description, and what I mean with that I come in a second, where this approach is now based on the genetic toggle switch, kind of a so standard tool there, and the motivation for that one is that all branching points can be considered as a binary cell fate. So there's also a longer story to that one, and in any case, so this would be then the differential equation that describes now your cell fate between an X and a Y state. So meaning like excitatory neurons or inhibitory neurons, right? Like a cell marker. And the genetic toggle switch is then based on the self-activation of a gene and cross inhibition. So meaning X is activating himself, Y is activating itself, and then there's a cross inhibition, where this cross inhibition is then, or is considering nonlinear behavior to consider, like for instance, saturation of your gene production. Okay, so very quick, what we can do now is that we can look into the phenotype space or the phase space for the physicist. So where we have on one axis the concentration of marker 1, so meaning that could be the excitatory and this is the inhibitory neuron, or the red and the white blood cell lineages. And then you can do a stability analysis of your system and look where are the stable points. So where would the cell end up? And you can see here the multi-stability. So if you have 4 different points that are actually then by changing the coupling of this interaction is then merging here, and you get then independence of this K parameter, you get then either one stable solution or triple or threefold solution between the cells can now change. As said, so we need also noise in this description, and I will skip this part here, but just to mention, so you can actually derive also analytical solutions for that to get a better understanding of it. But in terms of the rebellious cells, so we can now investigate this behavior in our phenotypic space by modeling, and can now start to say, or our starting condition is here that we now have a stem cell population localized here, and now we do the induction of one lineage. And then what you can see here, well, now you can see what's happening. So that actually based on this flow diagram, so there's one population that is directly going towards this this steady state, whereas the other part of the population needs a longer time, meaning first going into the wrong rebellious direction before eventually then reaching the induced cell state. So I will skip this as well here and come to my conclusion, where just as a catch up, so cellular heterogeneity can play an essential role in health disease transitions. So there is a broad spectrum of single cell analysis tools, so pipelines where, and I think it's important that you understand the technicalities if you analyze data, where it's coming from, so that you're aware of potential biases and issues that you have. Single cell analysis is still kind of a moving target, so where I introduced the currently consensus kind of tools with Surat, Monocle, and ScanPy, and still making the issue that these tools are now used for wide characterization of a lot of different systems, but actually to integrate this data and coming down to new mechanisms, this is, at least from my perspective, rather limited. So I mean, at the moment, it's really like the game to run just more samples without making too much integration and mining out of it. This is one I want to acknowledge my team, specifically Camille, who's running most of the experimental approaches. Dimitris is master head of the single cell analysis right now, and Sylvia has done most of the work here for the modeling. And I have to change the picture because Elle is not here on that one. Sorry, Elle. And then I thank you for your attention.
So thank you, Alexander, for this very nice lecture. It was also excellent introduction to single cell technologies. So I'm asking first, please, our ESRs, our students, if there are any questions from their side. Please don't be shy. Yes, please. Yeah, I don't know all the names yet. Maybe it's easier if you come here and ask the question. Might be easier, please. This one, you have to come on the stage.
Yes. Okay. Yeah, so thanks again for the lecture. I really enjoyed this introduction and also zoom into your particular field. And I just would like to clarify two issues. First of all, is the definition of attractor, because I'm familiar with this term from physics, and I have not seen any. Well, I'm just not sure I fully understand the meaning in a single cell field. And secondly, like as the first definition is linked to the terms of energy or like some kind of entropy, could you please describe which kind of energy do you, or like entropy do you measure, and what are the parameters from single cell data set which reflect or are the source of this energy? So 2 issues. Thank you.
Okay, so then in terms of the attractor, right? So I mean, the link there is that the Waddington epigenetic landscape is considered as a quasi-epigenetic landscape, meaning that the underlying mechanisms. So and this is then similar to the model that I've shown about the toggle switch, can lead to different steady states, meaning that you have multi-stability based on your dynamical system, right? So now adding the probabilistic perspective to it, so in terms of the, like for instance, the Fokker-Planck equation, we know that this around the steady state you will have a higher likelihood to be caught. And that is then actually the inverse to your epigenetic landscape. So I mean, that's a little bit the logic how physicists approach this epigenetic landscape, where I totally agree with the concern that this is actually an oversimplified perspective to it, because we do not have a real energetic, a real energy that we can assign to the transcriptional perspective, plus that so what this quasi-epigenetic assumption assumes that this is kind of given, right? So in that you just induce it, and actually the real process that is happening is self-organization, right? So I mean, each cell is in contact with the environment, is contributing to the environment. So that's why this epigenetic landscape isn't per se, or biologically, it's not a fixed structure, but a self-organized event. So that is then probably coordinated on a higher level. So and that actually is a part that I had to skip a little bit here, where we look also how does the phenotype distribution changes when we have population coupling. So meaning that this is also an indication that there is a feedback. Mm-hmm. In terms of, so how can single cell data help here, right, is that we are working on an approach where we actually use tools from non-equilibrium statistical physics. So to respect this non-equilibrium situation of the cell, and then you can then use their entropy production or entropy measures to try to clarify or to classify the different cell states. But this is first the early phase of the project. Yeah.
Thank you. And just like the recap, so is it about zooming into the particular subset of genes and their variability or the dynamics, or?
So it is. So I mean, what we have shown is that you can use actually this entropy-based measures to identify other other genes or driving genes of these of the differentiation process. But to really make a mechanistic link between real energetic cost of a cell to the epigenetic landscapes, this is totally underexplored, I would say, but we hope that with L we will make some efforts there to understand how actually the energetics of a cell is acting on cell differentiation.
Yeah, very interesting. Thank you.
Thank you. Can you just please introduce yourself?
Yeah, sure.
Maybe somebody.
Yeah, I'm Marina. I'm ESR 12 from UMCG Groningen. Yes.
Okay,
thank you.
And I think that there was another question from the ESR side. I clarified. Okay. So I forgot to ask the audience that they can put also the questions to the chat. Maybe I have, Alexander, one question regarding this first part that you actually try to avoid. Talking about the quality of the sample. So if we are talking about biological samples from patients or even from animal models, how critical it is that you get to the single cell analysis, to this workflow, fresh tissue? Are there any possibilities that you freeze in between? If you can elaborate a little bit on this.
Yeah, I think so. In the beginning, in the early phases, we were always concerned about frozen samples. So one issue there is that with frozen samples, that if you then start to put them into a droplet-based approach, so that we observed rather high amount of leakage. So, meaning my recommendation, if you work with fragile cells and frozen samples, I would mainly prefer to work on single nuclei experiments, because there you have a much better protection and at least in our hands a much higher reproducibility for the analysis. So if this is not possible because you do not have enough money for high coverage sequencing afterwards, you can. So I mean, there are optimized protocols now also for frozen cells, but again, there it depends pretty much which cell types you're working on. For brain cells, I have my doubts that you can actually go there very well with frozen samples and not doing single nuclei, but single cell. But for hepatocytes, for instance, so that's easier.
Okay, thank you. Hello, everyone. Welcome back from the coffee break. We are starting our second session today, and I would like first give the floor to our coordinator, Professor Christel van Steen, for a few words, and then we continue with the program. Please, Christel.
Thank you, Damiana. So yes, as a coordinator of TranSYS, I also welcome you very much to our second training school. And actually, the latest news is that we have over 240 registrants who showed interest in the existing program that was put together by the organizing committee, Benno Schwikowski, your host, Benham and Federico, 2 of our 15 ESRs, and the network coordination team. Unfortunately, I cannot be there with you, ESRs, in Paris. But be assured that I'm being regularly updated by snapshots from Christina or some ESRs. And I'm sure that someday, sometime, we will meet as one big TranSYS community with PIs and ESRs and our fan clubs. And I wish you all the best and very exciting time in Paris. Damiana, I give the floor back to you to introduce Laurence.
Thank you very much, Christel. We really hope that next time will be in vivo for everyone. Now I would like to introduce Dr. Laurence Collette. She's from the Drug Development Institute in Belgium. She's one of the fans or collaborators of the TranSYS consortium, and we are really looking forward to present your lecture. Please, the screen is yours.
Right, fine then. All right, so then without further ado, let's start. So it's really my pleasure to talk to you today about clinical trials. So I've worked 25 years now in this field, mostly in oncology, but I will try to give you an overview of the process of drug development and new treatment developments, not focusing too exclusively on oncology, really to give you a broad overview. So in general, what you need to know is that the time it takes to develop a new treatment and get it to the market, starting from preclinical and basic research and leading to the time when actually the drug is available in the clinics, is extremely long. And it goes into a number of phases. So we have preclinical research, which I think most of you have or will be involved with, when actually a very large number of compounds are being tested. And it's actually known as the Valley of Death because of all those compounds and potential treatments that are tested in cell lines or animals in the lab. Very, very few actually make it to even being tested in humans. So we see here a very, very large attrition rate, and the time for this phase is usually 3 to 6 years per compound. And then what I'm going to discuss with you today is the next stage of the drug development, which we call the clinical research, because it's in humans. And this, again, is made in a number of phases with different types of clinical trials. The very first ones that we call phase 1 are really to try to find a safe dose to give to humans. Once this is found, then the treatment undergoes a next phase of testing, which is trying really to see, to confirm the safety, but also to try to find in which disease this drug might work. And in cancer, that would be a specific type of cancer, or a specific subgroup of patients bearing a certain mutation, and also to see if there are signs of activity of this treatment. And then it's being tested in larger scale to really compare that new potential treatment to existing treatments and see if it brings additional benefits. And at that moment, the treatment, if it's a drug or a device, will be given authorization for marketing by regulatory authorities. In the US, it is the FDA. In Europe, it's the EMA, so the medical agencies. And then after that, there will be additional studies to confirm the safety of the treatments in once it's given in the clinic, and those are called phase 4 studies. But also in each country or region, there will be an assessment of the cost benefit of the treatments. And it's only once the authorities in a nation are given authorization to reimburse the treatments that generally the treatments are effectively made available to new patients and new patients not treated within clinical trials. So you see, it's a very, very long process. I just want to first come back to the definition of what is a clinical trial as opposed to other kinds of experiments. So there are a few standard definitions. So it's pretty broad. It's a research study that involves human subjects, where prospectively the patient is assigned to an intervention to evaluate the effect on health-related biomedical or behavioral outcomes. So it can be in healthy participants. It doesn't need to be comparative. It is anything really that has a component that would deviate from standard clinical practice. And it can also be PK studies or safety studies of new drugs. So it can also be an intervention that is not a drug. So really, it's a very broad definition. As you imagine, because these tests are done in humans, we cannot do everything we want. It is a very regulated field. And the basis of the regulation is really what is called the good clinical practice, which actually provides international ethical and quality standards for conducting clinical trials in humans. So it's actually a legal requirement. And the basis of these regulations are to ensure the protection of the patients and the well-being of subjects involved in trials. That's one aspect. And the second is to avoid waste, in the sense that it would also not be ethical to involve patients in an experimental treatment or an experimental intervention if this experiment is not conducted well enough so that the study is unlikely to give any credible results at the end. So these are really the two aspects of the regulations. And this is additional. In addition to that, we have specific regional regulations. And in Europe, we are really very strictly bound to clinical trial regulations and to all the regulations pertaining to data privacy, for instance. So really, those are elements that we need to take into account when designing studies and when conducting studies. So it is not strictly statistics or numbers or experimentation, it's also a lot of regulatory burden. So if I come back to the various phases of clinical trials that we saw in the former slide, giving them a little bit more details. So we have the phase 1 studies that really are to determine the dose of a treatment. So it's generally a small number of patients treated in a small number of institutions. And really the primary aim is to assess the side effects. Then you have the phase 2 that will assess early signs of activity. And these are called exploratory phases because these are really learning phases where we try to further define, for example, in targeted agents, the pathway of action and the actual assay that should be developed to assess the presence of a relevant biomarker, the population to be tested. And the phase 2 and 3 trials are the confirmatory phases of the efficacy of the drug, and those really are key to the drug development. So before discussing specific aspects of the early phase studies, I just want to quickly discuss the phase 3 trials because I will need some of the concepts to explain the phase 1 and phase 2 later on. So, so you know what we're talking about when we do a phase 3 trial. So that's the last step before putting a treatment to marketing authorization, or if it's an academic trial, to giving evidence to doctors that this treatment is better than an existing treatment. This phase 3 trial really aims to give insights into the risk and benefits. So it's a randomized trial, always, comparative, and the comparator is generally, it should be the best available standard treatment, or sometimes placebo control is considered when there is no existing treatment. It's in a large number of centers, usually in multiple countries, to ensure that there is sufficient representation of the treatment effects so that the results can be generalized. It's a very large investment, and it should be practice changing if it's positive. The primary measure in such trial should be really a clinically relevant endpoint, an endpoint that directly measures how the patient feels, functions, or survives. It should also be ideally a characteristic that is not subject to interpretation, so that we are certain that in the two groups being compared, if we speak, for example, of overall survival, it's very clear if a patient is dead or alive. There is no debate about this. This will be evaluated the same way in every country. However, if we were discussing an element of worsening of pain, for example, we may have influence of the different countries and how pain is perceived and dealt with in the various communities. So it should be a hard endpoint. And in terms of evidence, the best is to have this hard endpoint as your primary endpoint. But if it's not attainable, for example, if the disease is not lethal, you would have a validated surrogate for this endpoint. So in oncology, if we cannot attain survival, we would have progression-free survival, for example, as the endpoint. If you're in a cardiology disease, you may have cholesterol lowering, for example, as an intermediate endpoint. Some of which ones are accepted or not is also regulated and is subject to statistical demonstration. So the trial, as I said, is generally randomized. And why is it so? Randomization is then assigning treatments at random. The reason why we randomize is essentially because we want to be able to attribute any observed treatment differences in the endpoint to the differential treatment that is given to the patient. So the first thing is to avoid that leaving the doctors to decide which treatment to give to the patient would lead to non-comparable groups with patient characteristics being different and therefore expected outcome being different in the two groups. So for example, you could have younger patients and older patients, and then obviously the older patients would have worse survival, and you don't know if it's due to the treatment or to the age of the patient. Also, there are all other aspects that the patient undergoes during his management, during the time course of his life and treatment of his disease, such as assessments, general clinical care. And we want to have that as comparable as possible in the two groups. And this is why we assign treatments at random and why we have a protocol that specifies every other aspect of the conduct of the trial. But also only randomization, you could say I can make up groups that are similar in age, for example, or I can make up groups that are similar in disease extent. But you cannot make up groups that would be similar for yet unknown biomarkers that would be predictive for the outcome. So only randomization in large numbers will make, by the play of chance, the groups also comparable in terms of those unknown or unmeasured outcome measures. So far, randomization and randomized control trials, meaning with a protocol, are the best source of assessing causality. So for study design, how do we imagine those studies? We effectively have two types. We have the studies that show superiority of a new treatment, and we also have studies that show that the new treatment is almost as good as the existing treatment, but comes with other benefits, such as better quality of life, better tolerability, or an oral drug that is less invasive to patients than an IV drug because they can take it at home, etc. So the assumption about the underlying maths are really created without knowledge of the trial data, with the aim also to control the risk that we would make a false conclusion at the end of the trial. So we essentially base the calculation of the number of patients we need to follow on the basis of a simple statistical test, which is a test that there is no difference in the outcome between the two treatment arms versus the alternative, the one we want to show, which is that there is a benefit of the new treatment compared to the existing treatment. So we have a certain measure of this treatment effect. So it can be the hazard ratio or difference in proportion, depending on the type of endpoint that we consider. And then we will size the trial to have a certain probability of finding the effect if it's there, which we call the power, and control at the same time the risk that we declare that there is an effect when actually there was none. So typically a phase 3 trial will look like this one. That's an example of an EORTC trial that took a certain time to be published, but it's a good example. So it's simply a study in patients with rectal cancer. And the patients were assigned to two types of chemotherapies, one that involved oxaliplatin and one that did not. And the endpoint was to compare the progression-free survival of the two treatments. And the sample size was calculated with the idea that if we assume that without the oxaliplatin, there would be 65% of patients free of disease at 3 years, the aim was to show that we would improve that by 7%. And it was calculated that we needed about 1,000 patients for that. And the results will typically come with a test of hypothesis. In this case, the p-value was 0.861. And you see that in this case, it didn't work at all. And you see that there is actually no difference at all between the two treatments, right? The study was well conducted, but it shows that addition of this extra drug doesn't work. So the phase 3 studies hopefully are not all negative, but essentially the design comes like this. It's randomized with a comparison. Again, there are some constraints because we need to remain ethical. So there are interval reports of adverse events to regulatory authorities. There is generally a dedicated committee of experts that reviews the accumulating data of the study. It can be only safety or death, and that's to ensure that the risk benefit to patients is not in excess. Also in the view of emerging data from other studies, maybe, but also in long-term trials, particularly in oncology, you have to follow patients during years, and therefore you also have oftentimes a monitoring of the efficacy with the options to stop the treatment if things would actually not turn out positive, and that's actually what happened to the example that I showed, or to release the results if really there would be evidence that there is a treatment effect. Now, in doing that, sometimes you may have issues. For example, here, when you're testing, in this case, it's an immunotherapy, and you see that the benefit of the immunotherapy only comes after a certain lag time. So you have to be careful in the monitoring, because if you would look here at the data very, very early, you may take a false conclusion that there would be no benefit. So this is, again, a relatively complex thing to actually monitor the conduct of a phase 3 trial. But I will not spend more time on this. But really, I wanted to give you some view of how the phase 3 works, because that helps to understand how the other phases are conducted. So now, coming back to phase 1 and phase 2, I will start with how the phase 1s are conducted, and then how the phase 2 are conducted, and then give you an overview of where we're going now, because actually today all the phases are not really as separate as they used to be. So the phase 1 studies, it's the first stage of testing in human. There may be multiple types, though. It can be the very first time that a new drug is tested in humans. That's what we call a first in kind. So the first compound ever evaluated in humans for that type of, for that class of agent. It can be a first in human for the same class, but there was already some evidence for another drug in the same class. It can be associations of agents, or a new drug added to an existing therapy. But in all cases, the objective is to determine the recommended dose for the next studies and the future development, and to exclude the treatment if really there is no possibility to give it at a dose that is sufficient to have an effect, and yet to be safe enough. So the phase 1 studies really were developed with the vision that a higher dose gives more activity, and that's essentially what was done in, I mean, that was the case for chemotherapies, for example, but also for many other drugs. In oncology, because the disease and the drugs are, the disease is lethal and the drugs are very, very toxic, we cannot give, I mean, we don't enter in phase 1 patients that have still have therapeutic options available. So we generally take patients with a terminally ill disease who have failed all existing treatments and who are happy to test something new with the hope that things would help them a little bit. Because the patients are generally very advanced, we have issues of patients dying or failing the treatments very early. And actually, the response rate in phase 1 is overall about 5% of patients. And people are not paid for participating in phase 1. But in so in other disease areas, the treatments may be tested in healthy volunteers. in general, there is full control over how the experiment is conducted. It's generally conducted over a relatively short period of time. There is generally no individual benefit because the patient is not ill, so he would not or she would not benefit from the treatment, but there is generally a financial compensation given. So now, coming to how those studies are conducted, in general, the idea is to treat patients in sequence at increasing doses of the agent that is being investigated. The doses to be tested are based on feasibility and preclinical data, mostly. They start at a very low dose that is based on preclinical data, usually animal studies. So it's really 1/10 of the dose that would be lethal in dogs, for example, or in mouse. The increases between doses are generally not equal, so you would generally make smaller increments when you get close to the maximum dose. And the idea is to assess the safety. So we have a notion of what is called dose-limiting toxicity. It's generally a very important side effect. And this is observed during a relatively short period of time. So it's really what we call acute side effects. So the effects that occur immediately or very shortly after administration of treatment. And traditionally, the design would go enter 3 patients, look at the safety. Maybe I can show the traditional one would work like this. You would enter 3 patients, and then if you don't have toxicity in the first 3 patients, you just go to the next dose. If you have 1 dose-limiting toxicity, you add additional 3 patients. If you have no more toxicity, then you can escalate. If you see a second toxicity, then you cannot escalate anymore. Or if you have 2 in 3, you also have to stop. So the design really is very simple. You enter 3. If none, you increase. If 1, you add 3. If 2 in 3 or 2 in 6, you stop. It's a very popular design. It's easy to understand. It's easy to implement. It is still used a lot, so it's very, very simple and basic. The idea here is that the maximum dose is the one where there is less than 1 in 3 patients experiencing dose-limiting toxicity. The problems are that it doesn't allow to go back down. It lacks flexibility. You essentially have very few patients tested at your last dose level. You take decisions on 3 or 6 patients, so you don't have a lot of precision. And the study stops very quickly if you are, by bad luck, have 3 patients and for some reason, randomly, 2 of them have toxicity, your study would stop. So modern designs have been developed. Also, this design, it really uses only the last 3 or 6 patients to make a decision. It doesn't have memory of what happened before. So we have now more modern designs that allow to go up, down, and up again. So to retest the dose that has been tested. It's still cohorts of 3 generally, but other options are possible. And you can choose the toxicity level that you want. So it doesn't need to be 33% your target. You can say, I want to find the dose that is giving toxicity to no more than 20% or to, on average, only 20% of the design. And another feature that is very common is that once the highest dose has been found, the one that is considered for further testing, then additional patients will be tested at that dose level, which we call an expansion cohort, to try to confirm that that dose is indeed safe, also observing over longer periods, so not just 4 weeks or 3 weeks, but really over a longer period of treatment, and possibly also already seeing signs of tumor response. So for maybe the sake of time, I will not detail all the other designs, just to say that the newer designs, they generally involve modeling of a dose toxicity relationship and updating of this model after every observed patient so that you can fine-tune the assignment of treatments to the next patient. So for example, it would allow in this case, for this example, there is escalation to the third dose in one patient because there was no toxicity, and then de-escalation because there was a toxicity, and then because we had only 2 in 4 patients at this stage, we continue to give the same dose, and then it allows to re-escalate because overall you had finally not so many toxicities at the second dose level, and so it goes on like this. There are infinite numbers of such designs. The simpler ones are easy to implement in the clinics. The doctors don't need statisticians with them or scientists to AI people to run those models. And that's why they don't always go. I mean, why they're not really so much used in practice. So coming to expansion cohorts, I mentioned this. So we have dose level 1, 2, 3, say this is the one that is recommended, and then we treat additional patients. When it comes to treatments that are like in cancer, potentially could work specifically in certain types of cancers or in certain subtypes of cancers. Usually this part will include any type of disease, but then the expansion phases might be done specifically. For example, you take all types of solid cancers and then you do bladder cancer here, and then you do pancreatic cancer, lung cancer, breast cancer in each expansion cohort to better define also which is the population that you will need to use in the next phase of the development. So expansion cohorts lately have been pushed to their the extreme limit, and this thing happened here. I'm reporting one drug that was approved in 2014 for patients with non-small cell lung cancer harboring a particular mutation, and this approval was based on just the phase 1 trial. The phase 1 trial gave extremely positive results, and this treatment was essentially not treatable until then. But this phase 1 effectively included 59 patients. It was a huge phase 1 trial. Normally they don't take more than 30 patients. And the expansion cohort was 130 patients. So it's way larger than what normally is done in early phase studies. So I will come back to this trend a bit later. Coming now to phase 2 studies. So once we have done the phase 1, we essentially know pretty much the dose and regimen of the drug that we need to deliver. Then we have a bit of a better idea of what is the patient population that we want to evaluate. And in those phase 2 studies, there will be also a lot of translational research to better understand still the mechanism of action of the drugs. And we want to confirm the dose, to refine the population, and to try to evaluate already if this drug is a winner or if it's going to be a failure. So they shouldn't take too long. They shouldn't take too large numbers. And generally, you don't take in those phase 2 studies your final endpoints, such as survival. You would really more look into what does, so it's more a proof of concept that the treatment actually produces an effect in the body at the biological level that is as expected. So, for example, in oncology, one would look at the response at the level of the tumor rather than the progression-free survival of the patient, which are more clinical endpoints. In severe asthma trials, instead of the asthma exacerbation, which you need to watch for about a year, you would need to look at an earlier endpoint, such as, for example, the forced ejection fraction, which is, say, a predictor. Unfortunately, early endpoints are imperfect surrogates, so the belief is that there needs to be an effect on those endpoints in order to have a chance to have an effect on the final endpoint. But the fact that we see an effect on those early endpoints doesn't necessarily cause that there would be certainly an effect on the final endpoint. So there are still failures in phase 3 after the phase 2, but at least you exclude a number of drugs that actually do not work as expected, or do not work as anticipated, or do not work in the population where you thought they would work. So for phase 2, there is a plethora of designs, and I will give you all the binary options, and then we see all the possibilities. So there are two basic designs. So the single arm and the randomized comparative. So the randomized comparative, you understand why I discussed the phase 3 before, because effectively this would be a phase 2 that is built just like a phase 3. The only difference would be that you take another endpoint. So you have an earlier endpoint, a surrogate endpoint. In general, you would be looking for much bigger differences in that earlier endpoint than you would do on the final endpoint in the phase 3 studies, because you really need to see that there is substantial effect on that endpoint in order to believe that there would be an effect in the final endpoint, because the effects, the harder the endpoint, the longer term the endpoint, the more clinical the endpoint, usually the smaller the effect you see. And usually you would allow yourself maybe a larger error rate to make it possible to make a relatively small studies. So you may wish to give up on some risk of discarding a drug, or some risk of making still a phase 3 trial that would be negative. It's also essential to do a randomized trial when, say, you're in a targeted treatment in a newly defined subpopulation, and so you don't know what the response to your treatment would be in this newly defined population, because this marker was not tested, for example, in former series. Or you're using an endpoint that is so subjective, such as progression, that you don't have good reference data. In that case, you would need to have an internal control where you assess the same endpoint. The single-arm trials, essentially, they get rid of this internal comparator and they use a historical comparator. So they say in the past, with earlier series, we've seen that those patients have a 20% response rate, and then you would target your study to show that you have strictly larger response rate than what you know was attained. The problem with those is that you also have selection bias. So this population might be subselected. There may be a trend for better patients to enter or worse patients to enter. And also, if you define your entry criteria, those entry criteria might have changed. For example, if the imaging techniques were better now than in your historical series, your patient population declared metastatic would be different because the imaging is more sensitive, for example. The other approach that you find in phase 2, so one is the frequentist approach, so the same that we use in phase 3 based on statistical test of hypothesis. And as I just said, we would essentially do a one-sided test on the binomial response rate, for example, in the example I gave before, with 20% is what we achieved before. So we want to exclude that the response rate with the new treatment is less than 20%. We would determine something that is interesting, calculate a sample size, and then conduct a test at the type 1 error rate alpha and declare that there is an effect if our data are very unlikely to have been observed if the treatment really didn't work. And then there is more and more in phase 2 a Bayesian approach, which is essentially not based on decision per se in general, but is really based on, it considers that the true rate in your population is a random parameter, and it gives some prior view as to what is the true value of your parameter. So for example, your original view might be that you don't know what is the response rate, so you give equal likelihood of all the response rates in your population of interest. And then based on the data that you observe, you update your belief about the true value of the parameter. So in this example, having observed data that points in the direction of a response rate, I cannot read because it's too low, but I would say 30%, then you update your view as to what is the likely distribution of your true response rate based on your data. And in fact, Bayesian strictly would simply say, well, this is what I can tell you now about your parameter of interest, and then it's up to you to make up your mind whether you want to take a risk to go or not. Nowadays, there is more guidance given in general. People would have such kind of decision rules, like if after seeing my data, the chance that the true effect is greater than 40% is very, very high, or 0.9 or 0.95 or sometimes 0.7, you would just continue and consider that it is worthwhile. So it's really two different approaches to the analysis of the data with the same aim, which is to identify if there is a signal or not in the data that you've obtained. And last, you have studies with or without interim looks. So the idea in early phase 2 is really to stop exposing patients to a treatment that doesn't work. And in later phases, it may be that you have several doses of the treatment, or several still, because there is uncertainty, that's done mostly outside of oncology, or you have different treatments that you want to test in the same phase 2 for the same population. And then you would say, drop some treatments that don't work, or pick the one that seems to work the best. The danger, again, like with any interim look, is that if you release information about the potential study result during the course of the trial, that opens the door to more selection of patients, to more influence on your study. And so this needs to be done with a great caution. So there is an infinity of combination of types of designs for phase 2. You have single arm with internal control where you don't compare. So you have just a few examples here. Screening design. So here you screen a number of treatments for the same population and you pick the best one to go in the next phase. That's done by companies that have a number of treatments at hand and they don't have the capacity to test everything. You can have also for what is called basket trials. So it would be, say, a targeted drug targeting a certain gene mutation for cancer, and then you would have all types of tumors in the same study, but all bearing the same mutation. And then you would, in the same study, test either across all tumor types or in each tumor type separately. It can or cannot be randomized. You also have what is called platform trials. It's very common nowadays. So essentially all patients with a disease presenting with a disease would enter the platform. They would be tested for the presence of genetic mutations or biomarkers. And according to that, they would enter in certain parts of the study that would be specific to or targeting specifically a certain mutation. So I don't want to go into more details about those. It's a very, very interesting field, but we have limited time available today. So I just want to give you now a quick view of what's next. I think the trend now is that the phase 2 studies are often skipped or blended into other phases, and this is with the view of shortening the total treatment development time. So with the chemotherapies, it went to 10 years. With the molecular targeted agents, it went to 8 years, because the phase 2 came more as expansion cohorts. We've seen an example. And then nowadays, there is even another trend to give early approval, sometimes based on just phase 1 data. So how is it done? You can also have, so we have the phase 1-2, which are the expansion cohorts. We also have the blending of the phase 2 into phase 3 trials, where essentially you would have a first part of the trial that would consist in a randomized phase 2, and then an interim decision point, and then a continuation of the same trial as a phase 3. And it can be what we call operationally seamless. So essentially the phase 3 patients would be separate from the phase 2 patients. And from statistical considerations, there is no problem, no impact on the risk of, say, false positive conclusions. There are also what we call adaptive or inferentially seamless, where actually the phase 2 patients are also used in the analysis of the phase 3. And in this case, we need to take particular measures statistically to avoid the risk of false positive conclusions. There are trials nowadays that not only include phase 2 and phase 3, but include multiple treatments. So in this case, it's an example of a trial that continues to run in the UK. It was comparing treatments for prostate cancer patients. It started with 1 control group and 5 experimental treatments, and then different experimental treatments were added. The standard treatment even changed based on some results, but the platform is ongoing, and the patients, the comparisons are each time for the patients in one treatment with all the patients in the control group that were recruited during the same period of time. So these are really vast undertakings. So as we see, we were moving from this framework to really a framework where there is blending of the early phases into the confirmatory phases. So this is really blurred together with the view that regulators approve drugs. Nowadays, we've seen it for COVID, for example, based on earlier evidence, and require that there is confirmation of the effects in later phase studies that may confirm or not confirm, and that's why we have withdrawal of some drugs at some point. We had that with the COX inhibitors. There is a risk, of course, of skipping the phase 2, is that it's a very it's a massive learning phase, so it's a learning point that you skip. So there is a risk of having a phase 3, a greater risk of having to stop the phase 3 early. Now, in terms of expansion cohorts, so really approvals based on small single-arm trials. Generally, it's because it's really a very massive treatment effect for a disease where there is no available treatment, or for orphan indications, because in those indications where there are few patients, it's very, very difficult to conduct randomized comparisons. And in general, there is a requirement to supply additional data in a next phase. Another trend is the use of real-world evidence data. And there are some approval of drugs based on just real-world evidence, but not any kind of real-world evidence. For in this example that I take, it's a transplant drug that was approved, reflects a well-designed, non-interventional, relying on fit-for-purpose real-world data. It was a label extension, so it's not that this drug was never approved. It's approved, but for another disease. So it was approved for kidney, and now it was used already in the clinic for lung patients outside of the label. And there was a very good record of the data of all the lung cancer patients being transplanted in the US in a registry that was maintained in the US. There was also a very solid record of the death that could be linked, because in the US, they don't have the same data privacy control. There was a huge improvement that is beyond any bias that could actually cause a false positive result. And there is also additional evidence that the actual drug being tested is giving, making its action in addition to the other drugs that are given at the same time. In Europe and in the US, there are ongoing initiatives to try to understand how well real-world evidence studies could duplicate randomized clinical trials. So this initiative is really to try to see, can you replace potentially some randomized trials by real-world evidence? In Europe, we are a lot less centralized than in the US. So we have some registries, all are in separate countries. We have data protection regulations. So there is here a big initiative to try to put the EU at the level in terms of unified real-world data collection, so a repository and access to this real-world, and also a regulatory framework, because if you come to the regulators with real-world evidence, of course, they don't undergo the same data management, querying, quality verifications as trials, and currently, they're essentially used in post-approval or for label extension. We have that for the CAR T-cell, for example, there is a registry now, or when randomization isn't feasible. So in conclusion, I think I've shown you that trials are complex due to ethics, feasibility in the clinic, risk of biases, regulatory constraints, and costs. It's a long-term investment. The designs are complex, and I by far haven't covered all the aspects. They're essentially all rooted in two basic principles: to have unbiased estimate of the magnitude of the treatment effect and to control the risk of making false conclusions. Randomized trial remain so far the best source of evidence of a treatment effect. But not every hypothesis can be tested in a randomized trial. So I think in the future, we will see a combination of sources of evidence complementing trials. And this is just a picture of a paper published by the EMA team that showed that probably we will have some randomized trials in the first indications of a drug, and then we will probably go for indirect comparisons or real-world evidence when we will have to build evidence for additional combinations of these treatments or additional disease settings. But I will not detail that more, and it's actually more a view of the future. It starts to happen, but we are not yet there. So with this, I will stop. I think I've used more than my time, and I thank you very much for your attention, and I'm happy to take your questions.
Yeah, thank you, Dr. Collette. This was really a very interesting topic, a very interesting lecture, because we rarely see an overview what are the challenges of nowadays clinical trials. So I have to ask our local organizer, Professor Schwikowski, do we have a few minutes time for questions?
Yes, possible until maybe.
Yeah, so we will be exempt from this room. That's why I'm asking. Sorry. So please, ESRs, any raising questions from your side? Yes, please. Can you come here and ask? Introduce yourself, please. Okay.
Hello.
Hello.
I'm Catalina. I'm ESR number 7. And I wanted to thank you very much for your talk. It was very interesting. I was wondering, in those cases when you have already approved drugs, but you can test the combination, the efficacy, if they have synergy, for example, are in those cases the phases or the setting of a trial shorter or different somehow?
Well, it's a good question. It may be shorter or it may not be shorter. For example, if you already have somewhere information about the safety of the combination, because this was already tested in some hospital, or because you conducted that research, or you've already have patients treated with the combination in your hospital, then you might be able to skip the phase 1, for example. You oftentimes also would go more in the seamless phase 2-3. So you would have a comparator that would be the existing therapy, and you would add your new drug. The designs are a bit different if you replace a drug with a new one, but if you add it, you would have maybe a placebo in that comparison for that drug. So 2 drugs plus yours and 2 drugs plus the placebo. And potentially, if you have already a good view of the patient population, because it's the one used with the traditional treatment, you would blend the phase 2 into the phase 3, possibly, and have early stopping rules for safety. And then possibly early stopping rules for futility, and then directly go into the comparative phase 3. But if you don't have this safety, then you have to start from the beginning.
Exactly.
Yeah.
Thank you very much.
Yeah, so thank you very much again. I have 2 burning questions, but I cannot ask them now, so I will just leave them in the air. I would like maybe sometimes later your comment. What would be with statins if they would be discovered today? Because they would have likely much more difficulties coming into the market, but they are blockbusters now. So really, thanks a lot. I'm giving now the word to Benno.
Yes. Thank you so much, Damiana. Thanks a lot. Also from my part, I found it an excellent talk also for the occasion. Thanks a lot, Laurence. I also have burning questions that I won't even tell you now, but like me and everybody else also, I wanted to offer the possibility to send you these emails by these questions by email. Like we wrote in the chat of this meeting, please send them to my address, benno@pasteur.fr, and I'll forward them to Laurence, who may then be able
with
to
pleasure.
answer.
And if you have a general discussion session at the end, you may also make a sign and I could come back.
Okay. Oh, okay, cool. We might improvise on that too. Thanks a lot again. And
thank you. My pleasure.
For this talk. And I see all of you again in 1 hour at 1 p.m. here under the same link. Bye-bye.
Bye-bye.